Hello, everyone. In this chapter, we are going to address cancer and genetics. Now, why cancer and genetics? Because all cancers, regardless of what triggers that cancer, ultimately can be traced back to a modification that occurs at the genetic level. And again, regardless of what triggered that change. So, as we go through this chapter, a few things we want to keep in mind. It's one, to be able to define what the term cancer means, right? Explain how the loss of the cell cycle control can lead to cancer, and also how cancer can be inherited. Nevertheless, many, or if not most, of the cancers are not going to necessarily be inherited. What I mean by inherited, it's that we have intrinsic factors that predispose someone to cancer. We are going to define cancer cells. How are the cells behaving? And what is the role of stem cells in cancers, differentiated cells in cancer? Then we're going to dive into the genetics of cancer itself. What are the kind of modifications that happen at the genetic level so that a cancer situation may arise? What is the evolution of a cancer? What are the factors that increases chances of cancer, and what are ways to diagnose cancers, to treat cancers? So this is going to be the focus of our chapter. Now, let's first of all mark a pause with, not really mark a pause, but let's start with an introduction. And that introduction, I want it to be relatively inclusive. Inclusive in a sense that I want to give you as much information as possible in the introductory segment of this chapter. First and foremost, what is cancer? Well, cancer, it's uncontrolled cell division. Let me repeat that. Cancer, it's uncontrolled cell division. That is the basic definition of cancer. I expect you to know that. Now, understand that the cell cycle is tightly regulated in the body, because if the cell cycle is not tightly regulated, then the body can start see a multiplication of the cells without control and without reasons, which is a problem, right? Because these cells can now start not only cluttering the areas where they are growing, and as they clutter these areas where they are growing, they are preventing a proper functioning of the body. But in addition, these cells can break away from where they are growing and go to some other places in the body and cause some issues there. And typically the issues they are going to cause are steric issues. They occupy space and they cause a dysfunctioning of the whole system by being where they should not be at the first place, right? So I want you to remember that the cell cycle is tightly controlled. Now the question is, what controls the cell cycle? There are really three major aspects in the control of the cell cycle. We have genes, which job, when expressed, is to stimulate the cell cycle to go on. These are proto-oncogenes. The job of proto-oncogenes is to activate the cell cycle, to stimulate the cell cycle. So a cell can go from a G1 stage of the cell cycle to an S phase where the genetic information is going to be replicated. And from the S phase to the G2 phase, when the cell is going to prepare to enter the mitotic phase, right, which is the division, the distribution of the genetic information and the division of the nucleus, and eventually the cell is going to divide, right? Some examples of genes that stimulate cell division are the RAF gene, right? So that's an example of one gene. And then you have groups of genes that stop cell division or trigger apoptosis, apoptosis being programmed cell death. I want you to remember that. So these genes that stop cell division are called tumor suppressor genes. So they are going to cause cell division to stop. An example of a tumor suppressor gene is p53 gene. Now, if I was to take an analogy for genes associated with cancer, with cancer situation, the proto-oncogenes are like gas pedal in a car, right? So that when you hit the gas pedal, it's for the car to move on and to move faster and faster. Here is the thing. When you hit the gas pedal, if you never lift your foot off of the gas pedal, what is going to happen? Well, what is going to happen is that your car will never stop, right? Which is a problem, right? And that can lead to a crash. By the same token, the tumor suppressor genes can be compared to the brake pedal in a car. So tumor suppressor genes, when expressed, are going to stop the cell cycle. Just like when you are driving and you hit the brake pedal, the car should be stopping, right? What if you hit the gas pedal, the brake pedal, and the car is not braking, is not stopping? What would happen, right? Say you are on a highway. Well, what will happen is that eventually you are going to crash, right? So an alteration by loss of function of tumor suppressor genes is going to necessarily lead to cancer. Also, an overexpression of proto-oncogenes lead to cancer as well. And when proto-oncogenes become expressed overly, we don't call this proto-oncogenes anymore proto-oncogenes, we call them oncogenes. That I want you to remember. The last thing that you want to remember about origins of cancer, it's going to be DNA repair failing to repair properly. Let me repeat that. DNA repair failing to repair properly. That can also lead to cancer situation. So these three things, I would like you to remember them. Everything around, right? It can be environmental issues and other issues, right? Are going to be revolving around these three things: proto-oncogene turning into oncogene, tumor suppressor genes inactivating, and repair of DNA not happening properly. With that being said, let's move on. So, cancer, as I already mentioned it's an abnormal growth, right? An abnormal growth that leads to a mass of cells that we are going to call a tumor. So know that in the US, about 1 in 3 people are going to develop cancer. That may sound like a lot, and it is a lot. That is the reason why we have to be aware of the situation and start being ready for screening this type of potential disease. So, cancer, basically, the cell cycle is disturbed, right? So, we have some cancer susceptibility genes, right? Proto-oncogenes, tumor suppressor genes, and environmental exposure. These are the 2 things that are going to stimulate cancer development. All right. So the cellular pathway by which cancer is being produced, it's because of mutations in the genes, right? Now, these mutations are going to ultimately affect the cells. How so? Well, the cells can be affected in terms of their fate, right? Cells can technically should be differentiating, right? But this cell can lose the ability to differentiate, right? Or can even go backwards. So they differentiate. We are going to see this. Other aspect that is the consequences of the mutations is the cell survival. Now, the cells, as they multiply, they need oxygen, right? They need oxygen. And these cells, in order to receive oxygen, need blood vessels, right? So blood vessels are going to typically come and grow in oxygen, in cancer area, making oxygen available. But if oxygen is not available, the cells are not going to multiply, right? By not having enough oxygen, we're going to have a prevention. Well, actually, no, I'm sorry. With bringing oxygen and preventing apoptosis, cells will keep multiplying. Remember, apoptosis is programmed cell death. And then, in terms of genome maintenance, cells have to be able to maintain their genome, to repair their genome. However, if they are not able to repair their genome, what eventually is going to happen is cancer. So I did tell you about inability to repair and cancer. Then we are going to distinguish cancers based on really what is happening with the cells, right? What we call a tumor, it's basically a mass of cells from uncontrolled cell division. Tumors are going to occur in 2 kinds. We're going to have benign tumors. These benign tumors are mass of cells that grow in an area of origin and stay there in an area of origin. They do not invade other organs, right? They do not spread throughout the body. However, we have other type of tumors that are malignant tumors. These malignant tumors, because of blood vessels and lymphatic vessels, and because of the loss of these cells to attach to each other, these cells are going to actually go in circulation through bloodstream or the lymphatic vessels and be able to actually go and invade nearby tissues. And at that point, we say the cancer to be in a metastasis phases. In the metastasis phases, the tumor spread to other parts in the body, and at that point, it's nearly impossible to stop that cancer, right? And the cancer now can be lethal, so cause death, okay? And it's going to be very difficult to detect it, very difficult to actually treat it. Now, we have some conditions that can trigger cancer, and these conditions that are in general environmental are going to be called carcinogens. So carcinogens, radiations are carcinogenic. Some chemicals are carcinogenic. In any case, these carcinogens are going to lead to mutation in DNA, so DNA damage, right? DNA damage is going to be caused by mutagens. So mutagens are these carcinogens that cause mutations, right? Now, what are the different levels of cancers? Cancers are going to be classified based on which level we are looking at it and which level is happening, right? So we have at the whole body level, cancer can spread throughout the whole body, right? And when cancer spread throughout the whole body, there are a number of symptoms, right? We're going to have some biomarkers that are going to be present throughout the body so that you can actually isolate some blood. And from the blood, you are going to be able to notice the presence of abnormalities in the blood, right? We can look at some imaging to see a mass of cells in the lung, in the bones, or other parts of the body, and also mutation detection, right? And this mutation detection can happen by doing a biopsy of a part of the body. Then we have at a cellular level. So at the cellular level, we are going to have that some metabolic processes are going to be disturbed at the cellular level. So here we have the skin, and as we look at that skin, we have the epidermis right here, and we have the dermis right here, and we have the subcutaneous layer. Now, if we zoom in the epidermis, the basal layer of the epidermis, so what is close to the dermis, we are going to see that you have some cells here that are abnormal. The pink cells are abnormally dividing, while the brown cells are the normal cells, right? So here, the cell fate is going to be that the cells are going to be multiplying without control. They are going to lose the ability to differentiate into skin cells, right? They're going to gain the ability to go in circulation, which is not good, right? So we're going to notice that at the cellular level, we're going to notice this different aspect. Cells are going to become immortal, right? So they're going to be able to survive. And the genome is not going to be properly maintained. That's at a cellular level. But then we can take a look at cancers at the genome level. At the genome level, we are going to notice mutations, right? And these alterations of the genome can be the consequence of point mutation, right? For instance, we tend to see what we call a thymine pairing, right? So a thymine nitrogenous base is paired with another thymine nitrogenous base, right? This can actually, because of some radiations and other mutagens, this can cause multiple damages in the DNA. So the copy number variant can also change because of can change and actually lead to cancer. We have the chromosome rearrangements. What we mean by chromosome rearrangement, we talked about inversion, for instance. We talked about the moving of one piece of chromosome to another area of the chromosome or another chromosome, right? Aneuploidy, okay, which is too many or too little amount of chromosome. Change in gene expression. And change in gene expression, it's actually a major reason for cancer development. All right. So the cancer genes, again, I already mentioned this, but I'm going to repeat this. The proto-oncogenes, right, that turn into oncogenes. So when that happens, they are hyper expressed. Okay, there are about more than 100 genes that have been identified to be proto-oncogenes. So remember that proto-oncogenes are normal genes in the body, right? They are involved in stimulating cell division. However, what happens with these proto-oncogenes is that they overdo their job. And how do they overdo their job? By gaining function, right? So a gain in the function can cause cancer. A dominant allele that is going to be expressed can cause cancer. Tumor suppressor genes, right? More than 30 genes have been identified to be tumor suppressor genes. So any inactivation of tumor suppressor gene leads to loss of function, and that loss of function leads to cancer. Most of the time, this modification are going to lead to a type of allele that is recessive, which means that to actually develop a cancer with a recessive allele, you actually need two alleles that are modified, right? Because you receive one allele from mom, one allele from dad. So in as much as you have one of the alleles that is normal, you are pretty fine. But the other allele actually can remain silent, that's okay, right? However, because you only have one allele that is normal, what if that allele is mutated and there is loss of function? So this is when, being predisposed to cancer comes into play, right? And then we have DNA repair, which are really rare occurrence, but it does happen, right? All right. So we said earlier that cancer is because of uncontrolled cell division, which means that the cell division is controlled, right? So at which level that cell division is controlled and what controls that cell division? So we are going to have some protein growth factors that stimulate cell division. Now, by having too many protein growth factors in the body that are released without control, this is going to cause cells to multiply, right? Because these cells receive a signal that triggers cell division all the time. Then the other level, it's going to be the signaling molecule from outside, right? That actually are going to keep stimulating the cells to divide. Then we have the transcription factors from the inside. These transcription factors can be active all the time and causing uncontrolled gene expression, right? Which is going to cause overexpression of a gene. Remember, when proto-oncogenes express is to stimulate cell division. Now, if we have transcription factors that are systematically causing proto-oncogenes to express, well, that is going to lead to uncontrolled cell division. All right? So, going back to control in the cell cycle and the loss of control in the cell cycle, well, checkpoints are the places where the cells are verified if everything is okay to move on to the next stage of the cell cycle, or if something is at fault. If something is at fault, then the cell cycle is not going to move on, right? So this checkpoints have to be controlled tightly. So here we have a graphic representation of bone cells multiplying, and here we have the cell cycle. Now, at the G1 stage, the cell, a brand new cell, is going to check if DNA is okay, right? If there is no damage in DNA, the cell is going to allow to go on and do what it's supposed to do. If there is an issue with DNA that needs repaired, then the cell cycle is going to stop, right? Then, if everything is okay, then the cell is going to move to the S phase, right? Where DNA is going to be replicated. As DNA is replicated, well, the machinery of replication can make mistakes, right? So which means that we also have to check for integrity of DNA here. If there are issues with DNA, these issues need to be fixed. Therefore, the checkpoint is going to say, stop the cell cycle, right? So that this is repaired at the first place before we move on. And then we have the checkpoint right in the M phase, where the cell is going to check if all the chromosomes have mitotic spindle attached to the centromeres. Why? Because the new daughter cells, they all have to inherit the same amount of genetic information, right? So if the mitotic spindle are not properly attached to centromeres, the cell cycle is going to stop until everything is back to normal. But if it's not back to normal and that problem persists, whether it is a problem with DNA integrity or inability to properly distribute the genetic information, the cell is going to undergo what we call apoptosis, right? Or programmed cell death. You already know that term, right? So let's move on. So uncontrolled cell division happens because of the many things I already mentioned earlier, right? But remember, cancer is uncontrolled cell division. Now, in a normal situation, stem cells have the ability to renew themselves, all right? They also have the ability to differentiate into more specialized cells. Here, for instance, we have a neuron, some other specialized cells. Here, we have fibroblast, okay? Now, regardless of the differentiated cell, these cells can only multiply so many times, right? If you consider, for instance, the fibroblast can divide up to 50 times, which means that after 50 division, these cells cannot multiply anymore. they are going to undergo apoptosis and die. Why? Because of the telomeres, right? We talked about gene expression. We talked about the telomeres, right? So the telomeres are shrinking with the number of cell cycles, right? However, in a case of cells that are modified so that they undergo uncontrolled cell division, well, these telomeres do not shrink. Okay? Now, at that point, these cells are going to become immortal. So these are cancer cells. Okay? And these cancer cells are going to divide without limit. Okay? So the telomeres technically should be shrinking every single cell division, but in the case of cancer cells, these telomeres are not shrinking. In fact, these telomeres are increasing in length. Why? Because of telomerase. Now, during the process of development, there is a gene that encodes an enzyme called telomerase. Now, during the development, telomerase is active in embryonic cells. Why? Because the cells have to divide, multiply until the body reaches a perfect development. And then you have birth and the growth process. And at that point, from that birth, telomerases are technically not very active anymore, right? So the cells will now start dying off, okay? Dying off because of the limit as to how many times they can divide, okay? So I want you to remember these telomerases, and remember that telomerases are not produced all the time in our cells, right? Which limits the number of times cells can divide. But in cancer cells, there is no limit as to the expression of telomerases, which actually causes cells to undergo uncontrolled cell division. Now, there are some cells, cancer cells, that have been isolated by researchers. The cells are called HeLa cells. HeLa are actually named after the person from where the cells were isolated. Henrietta Lacks. Henrietta Lacks was a patient, right, from whom cancer cells were obtained. These HeLa cells are still used in laboratories today, right? This was in 1951. Now imagine how many times these cells have been multiplied, right? So these cells behave almost like bacterial cells that you can grow forever, right? So then we have, inherited cancers versus sporadic cancers. Inherited cancers, basically, this is written in genes, right? So the germ lines are affected and they are passed down to offsprings, right? And when germ lines are affected, the entire body is affected. So cancer can develop about anywhere in the body, right? While in sporadic cases, this arises because of a punctual mutation, a mutation that happens at a very specific spot, right? Leading to a cancer, right? So germline mutations are going to be passed down to offsprings. Cancer develops when a secondary mutation occurs. This type of cancers are rare, but tend to strike earlier in life compared to sporadic cancer. For instance, when we see children developing cancer, typically most of the time, it's because of germline cells that are mutated. Then we have somatic cancers. These somatic cancers are mutations that occur during the lifetime of an individual, right? Most of the time, when the body is starting to lose the ability to properly control the mechanisms that are happening in the cell, for instance, DNA repair. As we grow old, our ability to repair DNA decreases, right? So these are things that can lead to cancer. So the somatic mutations occur sporadically and they are not associated with sex cells, okay? Single dominant mutation or two recessive mutations, okay, which are very unlikely, right? Two recessive mutations are very unlikely to happen during the lifetime of an individual, but sometimes it does happen. And then we have cancer susceptibility is not passed down to offsprings. So here we have germline mutation, and here we have sporadic mutation that can lead to cancer, for instance, lung cancer here, okay? It's not passed down to offsprings, while germline cancer is passed down to offsprings. All right, so at a cellular level, the cancer begins always by a change in the genes, okay? And this change in the gene eventually are going to affect how the cell functions, okay? So if that cell that is not properly behaving is not identified as a cell to be discarded by our immune system, these cells will multiply and crowd the space and eventually move away from that area and go cause problems somewhere in the body. So now, what are characteristics of cancer cells? Well, characteristic of cancer cells, it's that these cancer cells typically are going to look different compared to normal cells in the area. So I showed you earlier the skin with skin cells that were normal, brown, and other skin cells that looked pink, right? But that was just a graphic representation, right? But we are going to see actually on the body what it looks like. So they look different from normal cells. That's something you have to remember. The second thing is that the cell surface is going to have different types of antigens or a difference in the number of antigens. So the quantity and the quality can be different as we compare it to normal cells. They contain mutations, right? Mutations that can be passed down or not, it depends, okay? The cells can actually move from one place to another. They can transplant because they can go in circulation through the lymphatic vessels or through the blood vessels. They can de-differentiate, meaning that they can lose their specialization, right? Which is a problem. They usually grow out of control, and they can invade other places in the body. So cells that are cancer cells do not know what we call contact inhibition. So in a normal condition, when cells are growing and they actually bump into each other, they stop multiplying. They stop multiplying because by coming in contact with each other, they are aware of the fact that the area is full. So that is what we call contact inhibition. But in case of cancer cells, they are not going to care about being against each other. They are going to stack on top of each other, right? By stacking on top of each other, they create a mass of cell, right? So they lack contact inhibition. So the cancer cells can move, so they can lose the ability to attach to each other and to actually be able to crawl to some other places, right? Which means that actually the cytoskeletal organization is affected in cancer cells. Metastasis is when cancer cells can go to other location compared to their area of origin, right? And with that metastasis, typically we are going to see angiogenesis. Angiogenesis is the formation of brand new blood vessels. What makes it necessary? The need for oxygen, the need for nutrients so that this cancer cell can receive nutrients and oxygen, right? Okay, you have a particular growth factor that is called the endothelial growth factor, actually the vascular endothelial growth factor, VEGF. This VEGF triggers the development of blood vessels in an area. So the other characteristic of cancer cells, it's that they're going to trigger the secretion of hormones, okay? And sometimes aneuploidy is going to develop having either extra chromosome or loss of a chromosome. Why would this happen? Because now understand that uncontrolled cell division, it's not just cell division that is happening out of control. The cell division are also happening faster than they should, right? By happening fast, in the process of distributing the genetic information, well, as the cells multiply in a quick manner, chances of failure in proper distribution of chromosome becomes high. Therefore, the possibility of aneuploidy among cancer cells. And you also understand that because of how quick the cells divide, you are going to have multiple types of cancerous cells. Yes, so one cancer cell may not be exactly genetically the same as another cancer cell. I'm going to show this to you when we actually look into the evolution of cancer. And you are going to see that even in one person, we can have multiple cancer cells that are different from each other genetically and based on the amount of genetic information. All right, so here we have a cancer situation, okay? So this is a cutaneous melanoma, okay? You can see it looks pretty nasty, right? And you can see that, for instance, the skin has a certain color here, but the color is different here, right? Look at the color here, and even here, it is different, right? So the cancer cells look different compared to normal cells. So when we do a tissue analysis, histology, so the tissue cells, so the cutaneous melanoma here are stained, right? And we can see the normal cells here in green, while the abnormal cells are right here in yellow or orange, if you will, right? They look different, they behave different, these cancer cells. Okay, so when we think of the origin of cancer, we are going to think of a number of things. Now, obviously, you have the genetic aspect, right, that we mentioned, but here we are going to address the cells, not the genes, but the cells, right? So, we're going to have activation of stem cells that can multiply so often that they crowd the area. We can have a dedifferentiation, so we are going from a specialized cell to a non-specialized cell that is going to undergo uncontrolled cell division, right? Increase in the proportion of a particular cell in a tissue, in its context, right? It can be a stem cell that increase in number compared to the normal number. It can be a progenitor stem cell, okay? It can be also, cancer can be caused by faulty tissue repair, right? So, more specifically, let's take a look at each of these situations. So, in this, we have that a cancer can come from a stem cell, right, that is going to multiply and slightly differentiate. They slightly differentiate, but keep the ability to multiply without control, right? We have that some of the stem cell can kind of specialize, right? But then eventually lose that ability to be able to control its cell division while that cell is specialized, right? Now, the cancer stem cells are going to need blood. So you have angiogenesis that is going to happen there. You're going to have cancer cells, stem cells that can develop in the brain. With angiogenesis, these cancer cells are going to grow even more, right? Cancer cell that can develop in the blood, okay? Well, obviously, in the blood, you don't have angiogenesis because the blood is already there, and the cancers are going to, just take advantage of all the oxygen that is in the blood to thrive, okay? We have the epithelium cancers, okay? Including skin cancers, cancers of the digestive tract, because the digestive tract, it's also a epithelium, right? The respiratory tract is also filled with epithelium, right? So these cancer cells can basically affect not only stem cells, but also specialized cells. So here we have stem cells that can multiply, self-renew, right? And this stem cells can actually develop into progenitor cells. Now we're going to have two types of progenitor cells: early progenitor cells and late progenitor cells. Now, the gene CD133 plus, it's a gene that is found in the cell. That gene actually can be lost at some point, okay? So in early progenitor cells, we have the gene CD133 plus, while in the late progenitor cells, we have the loss in the activity of that gene. That loss in the activity of that gene, it's a problem that actually ultimately is going to lead to uncontrolled cell division, right? So we're going to have, That is not correct. What happens is that this gene CD133 plus in late progenitor cell is lost. There we go. That gene is not expressed in late progenitor cells. There we go. And by the loss of that gene, this progenitor cells now are able to differentiate. There we go. They are able to differentiate into neurons, into astrocytes, which are supportive cells of neurons, and oligodendrocytes that are also supportive cells of neurons, right? This is in absence of CD133, in absence of CD133, okay? In as much as CD133 is present, the cells are going to remain progenitor cells. There we go. That's what I needed to explain. So now, in a cancer stem cell, the CD133 plus is there, right? Cells are going to be able to reproduce, right? So the cancer cells are going to divide and be able to self-renew, right? These cells eventually are going to become tumor cells, okay? And these tumor cells that are going to still keep CD133 plus is going to cause the cell to undergo uncontrolled cell division, while they should be differentiated, but they are not differentiated. Then we have the early progenitor cells. Normal, they have CD133 plus, right? These early progenitor cells, they lose CD133, which is going to now cause the cells to mature into different cells, so neurons, neurons, astrocytes, and oligodendrocytes. I'm not sure that that's the order, but that's fine, okay? Know that the cells are differentiating, which is what we expect, right? However, the early progenitor cells here can multiply, right, self-renew, and still keep the gene CD133 expressing. And in that situation, we are going to have a cancer situation. So this is from stem cells. And then we have dedifferentiation. In a dedifferentiation, we have a cell of the nervous tissue that technically is not expressing CD133, will actually go backwards. So they're going to turn into late progenitor cell and gain back the function of CD133. When they gain back the function of CD133, they turn into early progenitor cell and eventually to cancer cell and undergo uncontrolled cell division that way. Okay? Now we are going to address the proportion of cells in a tissue. So here we have a number of cells here with one cell that is a stem cell or a progenitor cell. So upon mutation of that one cell, right, we are going to have the cells that are going to increase in proportion. So we go from the top here, 2 out of 30 to now 5 out of 30. So the proportion goes from 6.6% here to 16.6% here. Now, upon another mutation, these cells are going to even multiply some more. Now we have 8 cells out of 30, which takes the proportion from 16.6% to 26.6%. Okay? So that is a mass of cell. That mass of cell represents a tumor. And then we have tissue repair. Now, in a normal condition, right, we're going to have epithelium here, say the skin, or the lining of the digestive tract, right? Say we have a damage in the skin, for instance, a burn, right? So you're going to have that these cells here, after injury, are going to renew, right? They're going to renew and they're going to cause a closure of this affected area. So as a consequence of that, we're going to have replacement of dead cell by brand new cells. So that's normal, okay? Now, when the tissue is constantly under attack in a chronic injury, what happens is that these cells that are coming to do repair, unfortunately, multiply without control, and eventually, out of these cells here, you are going to have cells that are going to mutate so that they develop into a mass of cells. So this is basically what can happen in uncontrolled tissue repair. In either case, you're always going to have a raise, right? You have a raise on the tissue. If we go back here, we have a raise on the tissue. If we go back here, you have a raise on the tissue compared to this area or even this area here, okay? So this concludes this first segment of this chapter. Thank you for your attention.